now that you've got a little background about Mendel and his work, we'll take a little closer look at how we use his fundamentals to help predict genotypes and phenotypes in organisms today. So we have to talk about probability before we can get into the genetics of this. So probability is the likelihood or chance that a certain event will occur. So usually when you learn about probability, we talk about flipping a coin. So the probability of getting heads on a single flip is 50%, and likely the opposite is true. The likelihood of getting tails is also 50% on a single flip. So when you flip a coin 2 times, each time has the same probability of 50% of getting heads. So just because you get heads the first time doesn't mean you will or you will not get heads the second time. They're 2 separate events. So the more times you flip the coin, the closer you would be to getting exactly 50-50. I mean, it's very likely in 2 flips that you could get 2 heads each time and not get the 50% chance to show up. And so this is where the more times you flip, the closer you're going to get to this 50% heads and 50% tails. So with Mendel, his pea plants for color, the heterozygote would be big B, little b. And so during meiosis, those homologous chromosomes line up during metaphase, and then they get separated during anaphase and go to different gametes. And so that's what we're showing here. We have that parent cell, and 2, since we end up with 4 cells at the end of meiosis, 2 of them will have the big B, and 2 of them will have the little b. And so half end up with the dominant allele and half end up with the recessive allele. And this follows the probability rules. So it gives you that 50% chance of having big B, and then you also have a 50% chance of having the little b allele. And so Mendel didn't know any of this, though, when he was doing his work. So during fertilization, a gamete with a big B or a little b allele has an equal chance of uniting with any of the gametes produced by the parent. So we use something called a Punnett square then to predict the genotypes and phenotypes of the offspring. And so the Punnett square is a chart for determining the expected percentages. Sometimes we actually use ratios as well of different genotypes in the offspring of 2 parents. So let's practice using the Punnett square. So determine the expected offspring of genotypes if these are our 2 parent genotypes. So a Punnett square is literally that, a square, and then you divide it into fourths. You write the parent genotypes on the outside, and it doesn't matter which parent goes on the top or which parent goes on the bottom. And then you fill it in. The B's on the top drop down, and then the B's on the left side slide across in its respective row. And so now we've completed our Punnett square, and we can see that for the homozygous dominant genotype, we have 1 of the 4 boxes. So 1 of 4 in terms of percent is 25% chance of the offspring will be big B, big B. Then you can see that 2 of the boxes have the heterozygous genotype, which is 2/4 or 1/2, which is equal to 50%. Then lastly, your last box is the homozygous recessive genotype. Again, there's 1 of the 4 boxes, which is equal to 25%. And so that's what helps us determine expected genotypes for offspring. Sometimes if you're asked to report the ratio, we would say that this is a 1 to 2 to 1 ratio. And for the ratios, you pretty much just count the boxes. So there's one box for the big B. There's two boxes for the big B, little b, the heterozygous, and then there's that one box for the homozygous recessive. So I think we have some more practice problems here. So a homozygous dominant tall pea plant is crossed with a heterozygous plant. What are the expected genotypic and phenotypic ratios or percents? So we need to figure out the parent genotypes first. And so if it's a dominant tall, you can pick which letter you want to use, but I'll probably use T for tall. And dominant is capital T, and it's homozygous, so that tells us they're both the same, so big T. And the second parent is heterozygous, so that's big T, little t. So then you can draw out your Punnett square. So we have big T, and big T, little t. And so we fill out the Punnett squares. And so our genotypic, we have two boxes here that are homozygous dominant. So that would be 50% big T. And then our phenotype, our second set of boxes is big T, little t. So there's two of those, so there's another 50%, and that's the heterozygous. So when we do that into a ratio, we have two boxes of big T to two boxes of big T, little t. But when you do ratios, they have to be in the lowest terms. And so the twos are in common, so we can divide each of them by 2 and get a 1-to-1 ratio. So you will need to make sure you keep your ratios in lowest terms. So then the next part is that it was asking for our phenotypic ratio. And so here we look at, all right, in this box, what is the phenotype going to be? Big T's tell us it's going to be tall. Same with here. This and these two down here, they're both the same. They both have a big T, so those two will also be tall. So 100% will be tall plants. Or in your ratio, it would just be a 1. So let's practice another one. Here we have a homozygous recessive short plant. So our parents and homozygous recessive short is crossed with a homozygous dominant tall plant. So draw out your Punnett squares and our recessive and our dominant. Fill in the Punnett square. And here for our genotype, they're all big T, little t. So we have 100%, and our ratio would be 1. And our phenotype, they all have that big T in them, which tells us it will be tall, and 100% will be tall, or the 1 ratio. So we can use Punnett squares to kind of work backwards. And so I think it's a white flower. And this side is for the purple because it has the big B. And so it's wondering or wanting us to determine that second allele on that parent. And so given this information, we know the little b has come from the white flower parent. And so given this information, we don't know enough to know what this allele would be. But if you looked at the offspring, if you physically had the offspring sitting in front of you and you could see, oh yeah, we have some white plants or white flowers, then you would know that this allele would have to be another little b in order to produce white flowers. You need that homozygous recessive genotype. So then that would tell you the second allele on the purple parent. And so we don't necessarily do a lot of these backwards ones, but know that it's a possibility. But the only way you can actually fill in your missing genotype is you have to be able to see the offspring. Otherwise, you'll never know. So what about a Punnett square when we have two characteristics? So up until this point, we've done the simple single, and we call this a monohybrid. A monohybrid cross. Mono for one. But now we want to look at two traits. They're heterozygous for pod color and pod form. So that means that one parent is big G, big F, little f. Actually, both parents are that. And so we need to figure out how to do a dihybrid cross. And it's dihybrid because we're testing two alleles. Di for two. So that means we have to make our Punnett square twice as big. So it'll be 4 by 4. And then to figure out the alleles to go across the top, we take one parent and we do all of the possible combinations of the G's and the F's. So we're going to take this first one, big G and little f is 1. And then our second one, starting with big G again, would have to now pair with the little f. Oops. And then you go, all right, our big G is paired with everything it could for the F's. Now we go to the little g, and it will pair with the big F. And again, it will pair now with the little f. So that's one parent. Now we have to do the same for the second parent. However, you notice that both parents are the same, so we're going to have the same possible alleles, and so we don't need to work them out separately. And so now, same process as before, but now instead of 2 letters, you'll have all 4. So this G came down, this G came across, this F came across, this F came down. So the G's, big G, big F, and now this one has little f. And we go across again, big G from the left, little g comes down, big F comes down, big F comes across. So when you're writing these, make sure that if you have the dominant allele with the recessive allele, the dominant allele always has to be written first, regardless of where it comes from. And so we'll keep finishing so that big G comes all the way down, the little f, big G, little f, big G, big F, little f, big G, little f. Third row, big G from the top, little g from the left side, big F from the left side, big F from up top. And keep going to fill this out. And lastly, big G from the top, little g from the left, big F from the top, little f from the left. So that would be how you fill out that dihybrid cross. And probably the trickiest thing is figuring out your parent alleles. But if you kind of do that distributive property kind of thing, it seems to work pretty well. So Mendel's work, he had to work backwards because he didn't know about alleles and genes yet. And so he used hundreds or even thousands of pea plants in each experiment. And his results were very close to the rules of probability. For example, his flower color experiment, he used 929 plants in the F2 generation. And remember, that generation is where we're going to have the 3-to-1 ratio, or 75% purple and 25% white flowers. And so of those 929 plants, 705 were purple and 224 were white. And so that's really, really close, as you can see, to that 75 to 25%. So when Mendel was doing his work, he only had the phenotypes to go off of. He didn't know about genes or genotypes, but he realized that there must be two factors that control each of the characteristics he studied, and one of those factors had to be dominant to the other. So his second set of experiment, he was pretty lucky when he was testing if traits were inherited together, like we just did that dihybrid cross, because the traits he picked were controlled by genes on non-homologous or different, this just simply means different, chromosomes. And so he never really had to deal with if the traits were on the same chromosome, would those characteristics still show up independently of one another. So with the advances in information we now know about genes, we know that inheritance is not as simple as it was in Mendel's pea plants. His pea plants were controlled by one gene that had two possible alleles. We had one trait that was completely dominant over the other, and it resulted in only two possible characteristics. And so geneticists now know that inheritance is way more complex than what Mendel had initially realized. So one of the exceptions or advances that we've discovered since Mendel's time is something called codominance and incomplete dominance. So codominance is a relationship between alleles for the same gene in which both alleles are expressed equally in the phenotype of the heterozygote. So this would be your example of codominance right over here. And you can see that we have red and white showing up together. And so when we have genotypes for a codominance, it's written in all capitals. So this would be like 2 big R's for red, 2 big W's for white, and then the red and white combo would be one of each. And so kind of key words when you see to know or to clue you in that you're dealing with codominance would be like spotted or speckled, somehow indicating that two colors are showing up. Incomplete dominance is the relationship between alleles for a gene in which one allele is only partially dominant to the other allele. So an intermediate, so that intermediate phenotype results. So this would be your red, again 2 big R's, plus your white, 2 big W's, gives you now pink. And again, your pink would have that RW genotype. And so here would be your incomplete dominance example, and we get the pink color there. And so in problems that deal with incomplete dominance, you would have to kind of look, maybe see the word blended. And so I always think of like mixing paint with incomplete dominance. And so if you mix red paint with white paint, you're going to get a pink paint. If you mix, yellow and blue to get green, or yellow and green to get blue, or whatever that is. Those are types of incomplete dominance. The other advancement is multiple alleles. Unlike Mendel's pea plants that only had 2 alleles, many genes will actually have more than 2 alleles, and your human blood type is an example of that because it has 3 possible alleles: the A allele, the B allele, and the O allele. And so the A allele, or the allele for type A, is codominant with the allele for the type B blood, and both A and B are dominant for O. So we have 4 possible phenotypes. You could be type A blood, type B, you could be AB, or you could be O. So if we had an AO genotype, we would know that we have a phenotype of type A blood, because A is dominant to O. So what genotype would you need in order to have O type blood? And so you would need to have OO in order to have O type blood, because O would be like your recessive allele. So this would make this the recessive allele. And in order to see the recessive phenotype, you need 2 of the recessive alleles. So polygenic characteristics are characteristics or a trait controlled by more than 1 gene, and each of which may have 2 or more alleles. So with polygenic characteristics, the inheritance is very complicated. You cannot do a Punnett square for them because they're so complicated. And so the results have a lot of different possible phenotypes. So like your skin color and adult height are both polygenic characteristics. So with these polygenic characteristics, the genes can be on the same chromosome, or they can be on those non-homologous or different chromosomes. So if the genes are located close together on the same chromosome, they are likely to be inherited together. However, it is possible for them to be separated during crossing over. However, the closer they are on the same chromosome, so here's a chromosome, and we have a gene here and a gene there, so let's call them A and B. Since they're relatively close together, they're less likely to get separated during crossing over, versus on this chromosome, if we had gene C and D, they're very far apart on the same chromosome, so it'd be likely that they could get separated during crossing over. So if the genes are located on different chromosomes or non-homologous, they may be recombined in various ways due to independent assortment. So again, not likely to be inherited together. So it's important to know that polygenic characteristics, they're very complicated when it comes to inheritance. So your genes are not the only thing that can affect your phenotype. So sunlight and nutrition play a role in a lot of our characteristics. So for example, if people in like developing countries, they have poor nutrition, it can prevent humans from reaching their genetically determined adult height. So if you're not getting good nutrients, you are not likely to reach the height that you should be because of your genes. And then exposure to ultraviolet radiation or sunlight can increase the amount of pigment in your skin, which makes it appear darker. And we experience this in the summertime, or if you go to tanning beds when you're outside all the time in the sun, we get a little tan. And so it changes our phenotype. The last thing is left-handedness versus right-handedness. We know that left-handed would be the recessive trait here, and right-handed would be the dominant. And so according to the rules of probability, there should be about 75% should be right-handed, and 25% of the human population should be left-handed. However, this is not the case. It's more like about 12% is actually left-handed. And this is because a lot of parents are right-handed, and so when their children are young, they unintentionally teach their children how to do things right-handed. It's simple things like putting a fork or a spoon on the right side of a child so that they just naturally pick it up with their right hand because their right hand is closer. Or even rolling a ball or putting a toy on their right side. And it's just things that adults do without even thinking about it. And so it's your environment can overrule some of your genetics. So there's about 13 or 12% of right-handed people who actually are genetically supposed to be left-handed, but that's just not the case.